Welcome to the Advanced Fluorescence Microscopy and Image Processing course. So today is the third lecture. So that is the first lecture of the second week. And today we will learn about design of a fluorescence microscope. So in today's lecture, I will show you how the microscope looks if you take the cross section of it, how the components are arranged inside the body of the microscope, and how different optical components are placed at different positions inside the microscope body. Okay, so let us start with the illumination paths. So there are two types of illumination paths: bright field versus fluorescence. So as we discussed in the previous class, that if you see the cells under the bright field illumination mode, you can see here the image of the yeast cells. So in the bright field, the kind of a school microscope, you can see only the external morphology of the cell. But using the fluorescence microscope, as shown in this figure, you can see the fluorescently labeled structures or the proteins within the cell. So here you can see that the tubulin is labeled with GFP, and the same cells which are shown here in the bright field mode, you can see those cells in the fluorescence mode. So how this both the light path differs inside the microscope. So for the bright field microscopy, you need the transmitted light path. So here you can see this is your illumination source, that is the bulb which gives the light. Then this is the condenser. Then you place your sample here and the light passes through the sample, and this is your objective lens. So objective lens collects the light from the sample and it makes the image either on the camera or on the eye. Okay, so if you see, this is known as the transmitted light path, because you see that light is actually transmitted through the sample and then it can produce the image like this. But in the fluorescence mode, it is known as the reflected light path, because the objective lens will not collect the light transmitting from the sample. Instead, it will collect the light being reflected from the sample. So how the light path of the fluorescence microscope works. So see here, the illumination source is here, the bulb is here, and bulb produce all 7 colors of light, white light basically. Okay, so white light is made up of all 7 colors. Now for seeing the GFP, you need a specific wavelength, that is 488 nanometer. So we'll put here the excitation filter. So this excitation filter will only allow the blue light to pass through. So out of 7 colors of the light, only the blue light will pass through this excitation filter, and it will be reflected through this dichroic mirror, and it will pass through the objective lens, and that light will illuminate the sample. And sample will reflect the light in all the directions, but out of all the directions, the reflected light which is in the direction of the objective lens will be collected back by the objective lens. It will again pass through the objective, it will pass through the dichroic mirror, and then it will pass through the emission filter, and then it will create an image either on the camera or on the eye. Okay, and that is known as the fluorescence image, and that looks like this. You can see any structures which is labeled with fluorescent proteins inside the cell. And here you can observe that this fluorescence light path is the reflected light path actually, and because of that the background remains black, because you are not collecting all the light getting transmitted from the sample. Instead, you are collecting only the light which is being reflected, and only the fluorescent proteins or the fluorescent structures which are there within the cell, they will reflect the light, and that light will be collected by the objective lens. So that these are the major difference between the transmitted light microscopy and the reflected light microscopy. And there are some other names. So this reflected light microscopy is also known as the fluorescence microscopy, or sometimes it is known as the epifluorescence or the wide field microscopy. Whereas this transmitted light microscopy is also known as the bright field microscopy, and it is generally used for seeing the external morphology of the cells. So let me show you. Generally the microscopes, the commercial microscopes, they are available in 2 configurations, which are known as upright or the inverted microscope. So here you can see the inverted microscope, and here you can see the upright microscopes. Now what are the difference between upright and inverted microscope? So if you see in the upright microscope, the objective lens are on the top of the sample. So these are the objective lens and your sample is here. So basically you are seeing the sample from the top. Okay, whereas in the inverted microscope, your sample is here and the objectives are at the bottom. So here you can see in this image, the objective lenses are at the bottom, and you will place your sample here. Now for most of the biological applications, people prefer to go for the inverted microscopes, because in the biological samples, like if you want to image the live cells, you have to put the liquid media on top of the live cells. So if the inverted microscope is there, the objective lens can see the cell from the bottom, okay, and on top of that the media can remain. Okay, if let's say if there is a media here, and if you try to focus the sample using the upright microscope, then the objectives cannot touch the samples, and because there is a liquid media here, it cannot focus the sample well. So that's why for most of the biological applications, people prefer to go for the inverted microscope, so that you can put the sample with the liquid media here, and from the bottom of the culture vessel you can image the cells. So if you see the light path, here both the light paths are shown, the transmitted light path and epifluorescence, or the epi-illumination, or the reflected light path. And all the fluorescence microscope, they are by default, they are equipped with the transmitted light path. Okay, so they generally comes with the transmitted light path, because first you have to see the cells, you just have to focus the cells like this, and after focusing you will switch on the fluorescent mode so that you can see the fluorescent signal within the cells. Okay, so see, in this inverted microscope, if you see the light path, this the red line represents the transmitted light path, and the blue line represents the epifluorescence or the fluorescence light path. So first let us see the transmitted light path. So here the bulb is here for the transmitted light, and that red color line, you can see that passes through the sample, and then here the objectives are there. So objectives will collect the transmitted light from the sample, then the light will pass through this route, and then it will create an image either on the camera or to the eye. Okay, now how the fluorescent light path work. So here is the light bulb, the white light bulb, which can emit all 7 color wavelength. So that is placed here, and from that light source, the white light enters through the microscope. Then here some filters are there, so that you can select the specific wavelength. Let's say for seeing the GFP, you will use the 488 nanometer wavelength. So that light will pass through the objective, and it will illuminate the sample, and sample will reflect the other color light, the fluorescent light, the green color light, that will be collected back by the objective, and that will pass through this route, and then you can see the image either on the camera or to the eyepiece. Similarly, in the upright microscope, if you see the light path, so the red line represents the transmitted light path. So here the the bulb for the transmitted light path is placed here at the bottom of the microscope body, and then the light comes through this route. It passes through the condenser, then it passes through the sample, then the light is collected by the objective, and that light will create an image either to the camera or to the eyepiece. And here the transmitted light path is here, sorry, the epifluorescence light path is here. The white bulb or the white light source is placed here. It emits all 7 colors wavelength, and then that light passes through this route, and here there are some fluorescent filters, so for the selection of the wavelength, and that fluorescent light will pass through the objective lens. It will illuminate the sample. The samples will reflect the light of different color, the fluorescent light, and that fluorescent light will be collected back by the objective lens, and then it will create an image either to the camera or to the eyepiece. So this is how these two different configuration of the microscopes, upright or the inverted microscope, works. And as I mentioned that any microscope, any fluorescence microscope is by default, it is equipped with the transmitted light path. So here I will show you the anatomy of the fluorescence microscope. So as I mentioned, here you can see the cross section of the fluorescence microscope. Okay, and this is the upright microscope, because you can see the objectives are on the top and the sample is here. Okay, so sample is observed from the objectives which are on the top, so that's why it is known as the upright microscope. So see, if you take the cross section, this is how the light path look like. So this is a mercury HBO lamp. So that is the fluorescent lamp which emits white color light, and see the white color light is coming all the way through this, and then here is a fluorescence filter. So from the white color light, only the green light is being passed from this point, and the green light will illuminate the sample. So if you see the slide, you will see that there is a green color light falling on the slide or the cover slip, and there is some fluorescent protein on your sample. Okay, that fluorescent protein will emit red color light. When you excite with the green color light, the emission will be in red color. So the red color light will be emitted by the sample, and that light will be collected back by the objective lens, and that light can go to the camera or to the eyepiece to show you the red color fluorescence. And you can see some other part of the microscope. So in this microscope, the transmitted light source is here, tungsten halogen lamp house. So the transmitted light source, light bulb, is here. It will emit the light, and that light will pass through this route. It will pass from the condenser. It will illuminate the sample, and it will be the light will be collected by the objective lens, and it will form an image either on the eyepiece or on the camera. Okay, so this is how the different parts of the fluorescence microscope look like. Now this is very important part. You can see at this junction you need basically three lenses or the three filters. One is the excitation filter. So what is the function of the excitation filter? So the excitation filter only allows one specific wavelength to pass through. So that you can select the filter and the wavelength. So let's say there is some red color fluorescence in the sample. So I need the green light for illumination. So I can put that kind of fluorescence filter which allows only green light to pass through. So the green light is passing through that filter and it illuminates the sample. Now sample will emit red light. So I want this filter in such a way so that the red light which is emitted from the sample can pass through this glass or this filter. So the red light will pass through this filter and it will be projected onto the camera or to the eyepiece. So we'll discuss this in detail in one of the next slide. So now let me show you there are several types of light sources available. So here I have shown the mercury HBO lamp, that is a fluorescent light source. But these days, because of the advancements in the technologies, there are several other options of the light source are available. And on this slide, I will show you that which kind of light sources are generally used in wide field or the confocal microscopy. So see, there are four types of light sources: LEDs, tungsten halogen lamp, mercury or xenon arc lamp, and lasers. Okay, so the one that we saw in this microscope is the mercury HBO lamp. Okay, so that is this kind of a light source, mercury or xenon arc lamp. Okay, but this is kind of a little outdated technology. These days, most of the microscopes, they are equipped with the LED light source, and LED is nothing but the regular. Generally, for several other applications, we use the LED bulbs, like that light emitting diodes. So there are several advantages and disadvantages of these light sources. So we will see those in this slide. So let me show you how this light source looks like. So here you can see the lamp house for tungsten halogen or mercury xenon lamp. So this is a lamp house for this kind of microscope. Here you can see this mercury HBO lamp is in a lamp house. So this lamp house looks like this. But if LED light sources are there, then the LEDs looks like this. And if the lasers are used as a light source, lasers looks like this. Okay. Now, why these days the microscopes have adopted LEDs over other light sources? Because the most importantly, you see the lifespan or the lifetime of these light sources. So LEDs can provide light for 10,000 hours, whereas the tungsten halogen lamp can provide light for only 2,000 hours. Every 2,000 hours you have to change the bulb, and these bulbs are really very expensive. In this mercury xenon lamp, the lifetime is only 200 to 300 hours, or the 400 to 600 hours for the xenon lamp. So every 300 or 400 hours you have to change the bulb, and these bulbs are very expensive. And lasers have a very long lifespan, okay, but they are again very expensive. Then another advantage of using the LED is the LEDs are available for different wavelengths. So let's say if you want red light or green light or orange light or blue light, you can find different LEDs. They are available in so many different colors. You can see Here I have shown different colors of LEDs from this manufacturer, which is known as the cool LED light source. Okay, and you see there are different wavelengths. The LEDs are available in so many different wavelengths, starting from 365 nanometer to 770 nanometer. So many different colors of LEDs are available. So they provide very specific wavelength of light. Whereas this mercury or xenon arc lamp, they provide the white light, and from the white light, you have to use different filters to select specific wavelength. Whereas in case of LEDs, you can directly get the specific wavelength. Okay, and another important point is here decay in the brightness across lifetime. So in LED, there is no decay in the brightness of the LED light. So as we discussed that the LED light can provide the light for 10,000 hours. So whether the LED light is new or it is old, even it is running at the 9,000 hours, the brightness will not change. The brightness will remain the same. Even you are using the new LED or it is already used for 9,000 hours. Okay. But in case of this mercury or xenon arc lamp, there is a decay in the brightness. So as such the lifespan is 200 to 300 hours or up to 400 to 600 hours. But the bulb is very bright in the initial few hours, but then the brightness uh, is reduced. Okay, so if you are using the bulb at around 250 hours, it will not give the same intensity of fluorescence as it was giving during the initial lifetime. Okay, so there are several other pros and cons which are mentioned here. You can just look into details. They are self-explanatory. Okay. Now here you can see uh, about the lasers as a light source. So there are different lasers and different gases are filled in the lasers for emitting different wavelengths. So see the argon ion laser provides a fixed wavelength of 351, 364, 457, 476, 488 and 514 nanometer. Whereas the helium neon laser, it provides a fixed wavelength of 543, 594 and 633. The lifetime of argon ion laser is 6,000 hours, whereas that is for helium neon laser is 40,000 hours. And the lasers also decay. Okay, as the mercury or xenon arc lamp, the brightness that the lasers provide in the initial hours is very high compared to the brightness that they provide towards the end of their life. Okay, so there is a decay in the brightness if you use the lasers. The lasers are not generally used for the wide field microscopy or the epifluorescence microscopy, but it has a specific application which is known as the confocal microscopy. So we are going to discuss more about confocal microscopy in one of the next class. So then you will understand that why lasers are used in a confocal microscopy. Because lasers uh, are lasers provides the high intensity monochromatic light that emits coherent and highly collimated beam of light. So what is the coherent light wave? So here you can see this is the coherent light wave and this is the incoherent light wave. So generally the lasers provide this kind of a coherent light wave, and also it provides the collimated light. So what is the collimated light? So this is a non-collimated light, or the unparalleled rays, it is known as non-collimated light, whereas the parallel rays, they are known as the collimated light. So lasers provide the collimated beam of light and coherent light source. So laser light beam can travel long distance without expansion, and it can be focused to a very small spot with a high level of brightness. So there are several bleaching applications that we'll discuss in some of the next classes. So when the bleaching is required, you have to focus the light to a small area on the biological sample. And for such kind of focusing, it cannot be done using the regular LED light source or the xenon or mercury arc lamp. Okay, for that we need the laser so that you can focus the light at very tiny spot on the biological sample. So for those applications, the lasers are useful and they are generally used as a light source in the confocal microscopy. And as we mentioned that there are different lasers, argon laser, argon krypton laser, helium neon laser, and helium cadmium lasers. So they provide different wavelength of light. There are other types of lasers also available which are known as solid state lasers. These days most of the lasers are solid state lasers. This is we are not using the gas based lasers because the solid state lasers have longer lifespan and they require small size compared to the huge size of these gas filled lasers. Then the dye lasers are also available, and semiconductor diode lasers are also available, and these lasers provides really long lifespan like 10,000 to 50,000 hours. But again, the price also goes up if you go for such kind of sophisticated lasers. So we will discuss more about these lasers as a light source while discussing about the confocal microscopy. So now let me bring you back to the fluorescence light source and the fluorescence filters and the dichroic. So as we mentioned that these light source that we discussed, the mercury or the xenon arc lamp or the tungsten halogen lamp. So these light sources basically provides the white light. Okay. Now how to select the specific wavelength for from this white light source? So for that, different fluorescence filters and dichroics are available. So what are fluorescence filters? So just to give you a simple example, you can see that here this red, green and blue, they are the fluorescent filters. So let's say if I pass all seven colors of light from here, red, green, blue, then only the red light can pass through, the other light will be reflected. Here this is a green filter, so only the green light can pass through, but the other lights will be reflected. Then this is a blue filter, so here only the blue light can pass through, whereas the other lights will be reflected. So these are known as fluorescent filters. So if you see the design of a microscope, you see here the cross section of the upright fluorescence microscope, and here you can see this is the mercury or xenon arc lamp, which is a white light source, and white light passes from this way, and there are several filter cubes you can see here. So the enlarged version of this filter cube is shown here. So these filter cubes are placed in the light path, and this cube contains three mirrors or the three filters. One is known as the excitation filter. This is the excitation filter from where the white light enters, and it will allow only one light to pass through. So from the white light, let's say this filter is for green light, so only the green light can pass through. Then that light will pass through the dichroic mirror, and it will fall to the sample. So and the reflected light from the sample or the fluorescence light will be collected by the objective lens here. So this is the emission filter. The reflected light will pass through the emission filter and it will be collected by the objective lens. So this filter cube is made up of three filters or the mirrors. First one is the excitation filter. Now what is the purpose of this dichroic mirror? So the unique property of the dichroic mirror is that, see, it reflects one wavelength of light, but it transmits other wavelength of light. So let's say you want to see the green fluorescent protein, for which you need blue light. Okay, so this dichroic mirror is designed in such a way so that it will reflect the blue color light that is coming from the excitation filter, and it will illuminate the sample, and sample will emit the green color light that will pass through this dichroic mirror, and it will pass through the emission filter, and it will also be detected by the camera or the eyes. Okay, so that is a unique feature of the dichroic mirror, that it reflects the excitation wavelength but transmit the emitted wavelength. And what is the function of this emission filter? So emission filter allows only the emitted wavelength to pass through and reach the detectors, and blocks the traces of the excitation light to reach to the detector. So the function of this emission filter, it only allows this emitted light to pass through and reach to the detector, but it will block all other wavelength of lights, so that you will only see the fluorescent signal with reduced background. So this is how these fluorescence filters, 6 or 8 fluorescence filters, are placed in the light path of the fluorescence microscope. And what are the wavelength you need? You just move this filter turret so that this filter cubes will move, and the appropriate filter cube will come in the right position in the light path, and it will excite some specific wavelength. So fluorescence, again, some more information on fluorescence filters and dichroics. So fluorescence filters are available in different types. Okay, so let's say the short pass filter. So as the name suggests, the short pass filter allows the lower wavelength of light to pass through, but it blocks the higher wavelength of visible light. Okay. Then the long pass filter. So that allows all the longer wavelength to pass through, but it will block the shorter wavelength. Then wide band pass filter. So if you see, so this is a wide band pass filter. So it allows most of the wavelength to pass through. And this is a narrow band pass filter. So narrow band pass filter blocks all other wavelength and allow only one specific wavelength to pass through. Now this is for the dichroic beam splitter. So dichroic mirror, they also are made up of such a material so that they can reflect this wavelength of light, the lower wavelength of light, but they can pass the higher wavelength of light. Okay, so these filters, fluorescence filters that you can design based on your application, there are several dielectric material available. So the glasses are coated with those dielectric material so that they can allow only specific wavelength to pass through and other wavelength to reflect. Let's say here is an example of the polychroic filter. So this polychroic filter, you can see it allows multiple wavelength to pass through. Okay, so depending on your applications, you have to select the filters in fluorescence microscope. Okay, suppose you want to see multicolor fluorescence, then you need this kind of a polychroic filter in this if you want to see multicolor in the same image. Okay, but let's say you want to image all the wavelength differently. Let's say one image for red color, one image for green color, one image for blue color, then you can go for this kind of a narrow band pass filter, so that first only the green light will pass and it will show the fluorescence, then only the red light will pass, it will show the fluorescence, then only the blue light will pass and it will show the fluorescence. Okay, so depending on the applications, you have to select the appropriate fluorescent filters and dichroics for your microscope. Now coming to the objective lens. So if you see the objective lens of a microscope, you will see so many details are written on the microscope, and all these details make some sense about the characteristic of the objective lens. So what it indicates? So see, first this is the brand name, Zeiss. Okay, there are mainly four uh, microscope manufacturers in the market: Zeiss, Nikon, Olympus, or Leica. So this is the brand name that this objective belongs to, Zeiss. Then this shows the magnification. You can see this is 63x. So 63x is the magnification of the objective lens. Also, this objective lens has this kind of different color codings. So let's say this upper line represents the magnification color code. So by looking at this blue color, you can see that this is the 63x magnification objective. If it is 100x, this will be a white ring. If it is 10x, it will be yellow ring. Okay, so based on the color, you can also identify the magnification. The bottom color line, which is red here, it indicates the immersion fluid. So let's say this is the oil or water or glycerin immersion objective lens. That's why it is red in color. If it would have been the oil immersion objective, this ring would have been black in color. If it is a water objective, it will be white. And if it is a glycerin objective, it will be yellow. So we'll discuss about in I think in last class, introduction to optics, we discussed that there are several different immersion fluids are being used with the objective lens, so that more reflected light can be captured by the objective lens to increase the resolution. So this indicates that this is the immersion fluid to be used with this objective lens. And there are some more information depending on your application. So here the contrast method, then mechanical correction collar for cover glass thickness correction. So some of the objectives have this kind of a knob to adjust the cover slip thickness. Okay, so and it also indicates which thickness of cover slip you should use with this objective. So the glass range of 0.15 to 0.19, so that is the thickness of the cover slip that you should use with this objective lens, because it depends on the working distance. Let's say some of the objectives which are generally provides high resolution and they have the higher numerical aperture, they have to be placed very close to the sample for observing the image. for observing the sample. Okay, so they need really thin cover slips, but some of the objectives uh, are long working distance objective. Let's say if you image the sample with 10x objective, so your sample will be here and your objective will be somewhere here. So there will be some enough space between the objective and your sample. So that doesn't need any kind of immersion fluid. Okay, but when you go for the higher magnification objective like 63x or 100x, your objective lens is almost touching to the sample, and at that time you need the immersion fluid. So if you see the real microscope, you will see multiple objective lenses. They are fixed uh, uh, on the microscope body, and by looking at this information, you can identify that this is a 10x objective. Also, from this color coding, you can also identify that this is 10x objective. This is 100x objective. This blue color represents 60x objective. Okay, so this microscope has 10x, 40x, and 100x. Blue color also represents 40x. See, so this blue ring represents a 40x objective lens. Okay, so by looking at this color coding, you can identify the objectives. Now coming to the detectors or the cameras used in fluorescence microscopy. So generally in the epifluorescence microscope or the wide field microscope, the cameras are used as a detector. Okay, and there are three types of cameras. CCD camera, that is known as charged coupled device. Then the EM-CCD camera, that is known as electron multiplying CCD. And SCMOS camera, that is known as scientific complementary metal oxide semiconductor detectors. Okay, so now the time now most of the microscopes are generally equipped with EM-CCD or SCMOS camera. But earlier, if you have any microscope which is maybe 10 years older, you may find some CCD cameras as well. And in the confocal microscopy, the photomultiplier tube is used. So that we'll discuss in more detail while discussing about the confocal microscope. So how the photomultiplier tube works. So briefly, I can say that. So the electrons which are coming from the samples, sorry, they are multiplied by this dianode and anode, and that's why it provides the amplified signal. So generally the photomultiplier tubes gives the amplified signal. So even if the fluorescence is weak, you can see it with the signal amplification. Whereas in this CCD and EM-CCD camera or the SCMOS camera also have this function, so which is uh, known as the gain. So by increasing the gain, this EM-CCD is the electron multiplying CCD. So by increasing the gain, you can uh, intensify your fluorescence signal. Okay. So generally, the EM-CCD cameras are good for the low light applications. Low light applications means if you really want to detect a weak fluorescence, then you should go for EM-CCD camera. Whereas SCMOS camera are generally used for the fast imaging application. So if you want to image your biological sample at very high speed, let's say 10 millisecond resolution, then it is recommended to go for the scientific SCMOS camera. Okay, but these days the SCMOS cameras have taken the market over EM-CCD because now now the SCMOS camera are available which are so sensitive like EM-CCD. So they provide both the advantage for uh, capturing the weak fluorescence as well as they can image faster. So these days the SCMOS cameras have taken the market of EM-CCD cameras. On this slide, you can see various types of sample holders available for imaging. So this is known as the LabTech 2 imaging chamber. It has multiple valves, so you can put your different cell types or same cell in different media with different treatment in the same chamber. Okay, so 4 different samples you can put in the same chamber. Then imaging chambers are also available. So if you want to see the cells in liquid culture, then you will grow the cells on this imaging chamber and you will put the liquid media on top of it. Then this is the regular slides and the cover slips, which are generally used for imaging purpose. Also, there are some microfluidic devices are also available. So for some live cell imaging applications, this kind of microfluidic slides are used so that you can pass different media, you can administer different drugs at different time points, and you can see how the cells are responding to different drugs. So these are the different types of sample holders. The most important thing is these sample holders are very expensive. And you will ask why it is expensive, because it looks like a simple plastic, right? But why they are so expensive? Because the bottom of these chambers provides the cover slip thickness. Okay, so generally the thickness of the cover slip is 0.13 to 0.16 mm. So the bottom of these chambers, they are made up of this cover slip kind of a very thin glass, so that you can even focus the sample with even high magnification objective lenses such as 63x and 100x. So that's why, because of the cover slip thickness, they are very expensive. Now coming to the fundamental problems in fluorescence microscopy. So there are four major problems that you will face when you will try to image any sample under the fluorescence microscope. The first problem is photo bleaching. Okay, so what is a photo bleaching? So let's say in this image you are seeing some red fluorescence and here some magenta colored fluorescence. Okay, now this fluorescence is not like long lasting. Let's say if you keep the microscope on and keep on observing this fluorescence for, let's say, 1 hour, it's not that it will remain for 1 hour. It will give the fluorescence signal for 1 hour, but instead it will start bleaching upon longer imaging. So you can see this is the first first minute, then this another image was taken at 25 minutes, the another image was taken at 49 minutes. So you can see the bleaching. Okay, the fluorescence signal is bleaching because these dyes have some specific amount of photon. So that photons are, once they deplete, then they cannot give further fluorescence. Okay, so this photo bleaching is an important problem in while doing fluorescence microscopy. So here the red light shows the fast photo bleaching, whereas the magenta dye shows slow photo bleaching. So let's say if your imaging application is like you want to image the cell for a long time, let's say for 24 hours, and you want to take the image at every 15 minutes. Okay, then you will use this kind of a fluorescent protein which shows so slow photo bleaching. You should not go for this kind of fluorescent protein which shows you which gives you fast photo bleaching. Okay, so when a fluorophore loses its fluorescence due to damage induced by the light, that phenomenon is known as photobleaching, and this leads to loss of fluorescence while imaging the sample. So how to reduce this photobleaching? So there are several ways you can reduce the photobleaching. So first thing is reduce the intensity of the excitation light. So let's say you are using LED for the illumination. Okay, now you can run the LED at 100% power also, and you can run the LED at 20% power also. So if you can see this kind of fluorescence from a fluorescent protein or a fluorescent sample with 20% power of the LED, you will not use it with 100% power. Okay, because if you switch on the LED with 100% power, it will bleach the sample faster. But if you can see the same fluorescence at 20% light intensity, why to go for 100%? So the first thing to reduce photo bleaching is reduce the intensity of the excitation light. The second thing is reduce the time of exposure to the excitation light. So as you know, in camera, when you take any picture, you adjust the exposure time. Okay, so if let's say you have to do imaging in dark, then you will use high exposure. When if the sample is very bright, you will go for the low exposure time. So if you can image this kind of structure with, let's say, 50 millisecond exposure time, you will not go for 500 millisecond exposure, because that will lead to more photo bleaching. So if you can see the structure with 50 millisecond exposure, just set the exposure time of your camera to 50 millisecond. Don't go for 500 millisecond to create a lot of photo bleaching. Then the third thing is use anti-fade reagents. So commercially, some reagents are available which are known as anti-fade reagent. So if you mount your cells in that anti-fade reagent, then the bleaching will be slowed down. So here you can see that in 25 minutes the fluorescence is bleaching, right? But let's say if you have put the samples in that anti-fade reagent, then this bleaching will be slowed down. So maybe you can see this kind of signal up to even 25 minutes. Okay. Then use fluorophores with low bleaching rate. Instead of GFP, use neon green. So green fluorescent protein bleaches faster. So scientists have developed the modified version of green fluorescent protein, which is known as neon green. And neon green has a high photostability. So it bleaches at a slower rate than GFP. So if your imaging application needs long hours of imaging, then label your protein with neon green instead of EGFP or the green fluorescent protein. Then reduce the oxygen in the sample. So this is one of the important factor that creates photobleaching. The presence of oxygen creates some kind of a free radicals, which are those free radicals can create this photobleaching. So to avoid that thing, you can just keep your sample in nitrogen instead of oxygen, or some oxygen scavengers are added to reduce the photobleaching. But you have to make sure that your application or your cells do not need oxygen for the regular growth. If you are working with the fixed cells, then you can use these oxygen scavengers, so they will reduce the oxygen from the sample and it will reduce the photo bleaching. And there are some mathematical photo bleaching corrections algorithms are available. Sometimes also they are known as deconvolution. So those kind of mathematical algorithms you can use for correcting the photo bleaching. So this is one of the major problem in fluorescence microscopy. Now coming to the second problem. So the second major problem in fluorescence microscopy is known as the autofluorescence. So autofluorescence is the natural emission of light by biological structures such as mitochondria and lysosomes when they absorb light. Okay, so let's say in your cell there is not a single protein which is labeled with any fluorescent protein, but still you are putting those cells under the microscope and then you are seeing this kind of fluorescence. So this is known as the autofluorescence. So here in this image you can see only 4 cells you are seeing, but actually there are multiple cells present. Only these 4 cells that you are seeing, they are dead cells and they show a lot of autofluorescence. Okay, whereas the live cells, there is no fluorescent label in it, so they do not show any fluorescent signal. So this thing happens. Let's say your sample is not labeled with any fluorescent protein or any fluorescent dye, and you are just putting it under the microscope, and you will see that some cells are giving you fluorescence. So that is not the real fluorescence. That is the autofluorescence, and generally the dead cells gives you autofluorescence because of the accumulation of a lot of secondary metabolites. Okay. And how to identify that this is an autofluorescence? Let's say you are seeing these 4 cells very bright or fluorescent under the DAPI channel, right? Now you will make sure that is not some kind of a real fluorescence. So you will check this sample in other color light, like with green GF under the GFP channel, under the TRITC channel, under the Sci-Fi channel. So this autofluorescence is generally visible in all other channels. Whereas if this would have been the specific labeling, then it will not give you fluorescence in any other channel except the blue channel or except the green channel. But autofluorescence is generally visible in all the channels. So how to overcome these autofluorescence issues? So there are several ways. The first one is sodium borohydride treatment can significantly reduce autofluorescence in red and green wavelengths. Okay, so you have to treat your samples with sodium borohydride to reduce the autofluorescence in red and green channel. Then the background autofluorescence can also be quenched with Sudan black bead dye or the photo bleached away with the sunlight or strong UV light. So sometimes you just have to expose your cells to UV light or the sunlight so that the autofluorescence will go away. Okay, it will be bleached. The autofluorescence itself will be bleached. The third option is amplify your real signal over autofluorescence using signal amplification kit. So some commercial kits are available from Invitrogen or there are maybe several other brands which amplifies the real signal. So let's say here in these cells you have some structures labeled with GFP, and because of the autofluorescence you cannot see them in a distinct way. Okay, so what you will do, you will use the signal amplification kit. So the signal amplification kit will amplify your real GFP signal over the autofluorescence so that you can see your real signal over the autofluorescence. They do not use phenol red in imaging media or use cell culture media for fluorescence imaging. So generally you must be aware that the cell culture media generally comes with the phenol red dye as a pH indicator, so that you can see that whether the pH of the cell culture media is maintained or not, right? So generally the media with phenol red should be avoided for fluorescence microscopy because phenol red itself can give you some fluorescence. So there are some cell culture media available for fluorescence imaging. They are colorless, and you should use such kind of media for imaging. Otherwise, you will see a lot of autofluorescence from the dyes such as phenol red. And the other way to overcome the autofluorescence issue is the software-based subtraction of autofluorescence, which is also known as spectral deconvolution. So as I mentioned previously, in case of photo bleaching, there are some computational algorithms are available which are being used to subtract the autofluorescence from the real fluorescence. So these are the 5 ways by which you can reduce the autofluorescence. Another important problem in fluorescence microscopy is the bleed-through effect. So what is a bleed-through effect? So it is also known as crossover or the crosstalk. And this can occur in experiments that involves labeling with more than one fluorophore at a time. So let's say this is a sample that you have labeled with 3 different colors: channel 1, channel 2 and channel 3, blue, green and red color. 3 different proteins or 3 different cell organelles you have labeled with 3 different color dye. But these dyes are not spectrally very well separated. So what will happen when you are seeing the blue color, you are seeing the structures well, but when you are observing the green color, you are seeing the green structures along with some bleed through fluorescence from the blue color, because these two dyes are not spectrally separated. Now what do I mean by spectral separation of the dye? So let's say for two color imaging, you can use these two dye also, like AF488 and AF555, and you can use these two dyes also, AF488 and AF633. So you see how much is the spectral separation is there between these two dye. If you use these two dye like AF488 green and 633, there is a huge spectral separation. So if you excite this dye, it will not excite the 633 nanometer dye. Okay, but here you see both the dyes are very close in terms of their spectra. Okay, this is 488 nanometer and this is 555 nanometer, and they have some overlapping area. So even if you are exciting this dye, the 488 dye, it will slightly excite the 555 nanometer dye as well. It will give you the fluorescence in the other channel as well. Okay, so when you have to do this kind of a multicolor imaging, you have to make sure that your dyes are spectrally separated. Whatever the colors of dye that you use, you have to make sure that your dyes are spectrally well separated. If they are overlapping, then you will see this kind of a bleed through effect. How to reduce this bleed through effect? So the first thing is reduce the exposure time to minimize the effect. So let's say if you are seeing this kind of structure, the blue color structure in actually the green channel, what you will do? You will reduce the illumination light for this green color so that it will reduce this bleed through effect. Second thing is use highly specific filter set with narrow band pass. Okay, highly specific filter set with narrow band pass. So while discussing about fluorescence filter, we discussed that there are different types of filters available. This is a narrow band pass filter. So let's say my two dyes are spectrally overlapping, then I should use this kind of a narrow band pass filter so that only one particular wavelength will illuminate one dye. But if I go for this wide band pass filter, then as such, it can illuminate all different colored dyes simultaneously, and it will give me the bleed through effect. So this is how you should select your filters and the dyes for imaging. And the third option is use such pairs of fluorophores to avoid spectral overlap. So as I mentioned, let's say if you want to see two colors and you have the option to go with 488 and 633 nanometer, you should go for it. You should not go for this kind of a combination of dye like 488 nanometer and 555 nanometer, because they are spectrally overlapping. Okay, so these are the three major problems in fluorescence microscopy. With this, let us end this class, and our next lecture will be on fluorescent proteins, organic dyes, and protein labeling strategies. Thank you.